Today we discuss about the complement system that comes under the serology and immunology topics. So first we discuss about that what is the complement system and who discovered it, what is the history of the complement system. So first we discuss about the history. Actually, complement system discovered in 1894 by the dead scientist. It represents lytic activity of the fresh serum. Which activity it represents? It represents lytic activity of the serum. Actually, there are three types of complement system. That is, first is classical complement system, second is alternative, and third is mannose binding lactin type complement system. In complement system, what happened? Actually, in complement system, they phagocytose or lyses the pathogen that enter in our body with one series of the one type of series of a chemical reaction by which different types of complement protein will be activated, and then phagocytosis or opsonization will be occur of that pathogen in this process. What? Opsonization. What happened in this? In this, a series of complement protein will be activated. Series of complement protein will be activated in this process. Okay, it represents lytic activity of serum, and its lytic activity destroy when heated at 56 degrees Celsius for 30 minutes. Because it's a high temperature, when we heat at a high temperature, the pathogen will be destroyed at that temperature. So this is the history of the complement system. Next, what is the function of the complement system? The main function of the complement system is the opsonization to enhance phagocytosis. Already I discussed in previous slide. The main function of the complement system is the opsonization. What? Opsonization to enhance phagocytosis. Another, phagocyte attraction and activation. What happened in this? When series of complement protein will be activated, series of complement protein. When this protein will be activated, they lyse the pathogen. What? They lyse the pathogen or infected cell. Regulation of antibody responses. They also regulate the antibody response. Clearance of immune complexes. Clearance of apoptotic cell. The cell which are apoptotic in nature, by complement system, they remove from the body. What? Remove from the body with the help of complement system. Next, protein of the complement system. What are the protein of the complement system? Actually there are several protein in complement system. In the sequence of the protein by which activated are different in different pathway, but there are several number of complement protein. The protein will be indicated by like C1, C2, C3. This is the name of the protein. The number is indicated the sequence by which they are discovered. The number what indicated? Number indicate in which sequence they are discovered. Means C1 is discovered first, after that C2, after that C3, like that. Okay. Another protein is factor B, D, H and I. These factor B, D, H and I. This protein or factor, actually there are three types of complement system, right? First is classical, right? Second is alternative, and third is mannose binding protein. Mannose binding protein. Okay, these are the three types of complement system pathway. So this C1 is in classical pathway, this is alternative pathway, and this is mannose binding pathway, helping mannose binding pathway. So mannose binding lectin, MBL associated serine proteases, this all are helping where? In third pathway, that is mannose binding pathway. And last one is C1 inhibitor, C4 binding protein, decay accelerating factor, complement receptor, and protein S. What? C1 inhibitor, C4 binding protein, decay accelerating factor, complement receptor. Now, what is the definition? What is the definition of the complement, and what is the activation of the C protein? How activated? So, C activation, alternation of C proteins such that they interact with the next component. How the C protein will be activated? They activated by a alternation of C protein such that interact with the next component. Next is C fixation, utilization of C by antigen antibody complex. When C protein will be fixed, then they are utilized by as an antigen antibody complex. Next is hemolytic units, dilution of serum which lyses 50% of standardized super suspension of antibody coated RBC. C inactivation, denaturation of an early C component resulting in loss of hemolytic activity. Clear? And last one is convertase and esterase. Altered C protein which act as a proteolytic, they act as a C component. They act as C component. Now, activation products of complement protein. Activated components are usually overlined. Already know they are C1, C2, C3. When they react, they converted into a first of all they are inactivated C1. Remember that thing. When they activated like this, a barcode is or line is present. This means they are activated. The activated components are usually overlined. Example C1QRS means they are activated. Clear? When enzymatically cleaved, the larger moiety binds to the activation complex or membrane, and smaller peptide is released in the microenvironment. Remember one thing, when suppose this C1, when they activated, they converted into a two fragment like C1A and C1B. Okay, C1A and C1B. B is a larger fragment. What? B is a larger fragment and A is a smaller fragment. This larger fragment bind to activation complex. Activation complex. Whereas smaller fragment released in the microenvironment. Means there is no use of a smaller fragment and there is used of this larger fragment. Okay, remember this. A is a smaller fragment and B is a larger fragment. But there is one exception, that is C2. What? C2. For only C2. That in C2, B is the smaller fragment and A is the larger fragment. Remember, this is the exception only. What? This is the exception. When enzymatically cleaved, the larger moiety binds to the activation complex or membrane and the smaller peptide is released in the microenvironment. Already I told you this is usually added to the larger membrane binding and peptide and A to the smaller peptide. But there is an exception, that is C2. In C2, the larger membrane binding moiety is C2A and the smaller one is a C2B. This is only for the C2. Remember this. This is the exception. So now, what is the pathway? What is the pathway of the complement activation? Already I told you that there are three pathways of the complement activation. First is classical pathway, second is Mannose binding pathway, and third is alternative pathway. In classical pathway is the antibody dependent pathway. What? Antibody dependent pathway. Classical pathway. And lactin and alternative pathway is the antibody independent. Remember this one, very important. Which is antibody dependent pathway and which is antibody independent pathway. So classical pathway is the antibody dependent and lactin alternative pathway is antibody independent. That when complement system is activated, activation of C3 and of C5 convertase. In each and every pathway, there is generation of C5 convertase. Very important. What? Generation of C5 convertase. When C5 convertase activate, they activate the C5 complement protein. They activate the C5 complement protein. After activation of C5 complement protein, this C5 complement protein help in lytic attack pathway. What? Lytic attack pathway. The opsonization of the pathogen by phagocytosis, so that the pathogen or foreign particles that enter our body will destroy. Clear? Which is antibody dependent pathway and which is antibody independent pathway. So now component of the, now we study about the classical pathway. What is the component of the classical pathway first? So the component of the classical pathway is the C1, right? C2, C3, C4. Clear? C1 protein, C2 protein, C3 protein, C4 protein. This is the C1 complex. The complex in C1 is in C1 complex, which ingredient is present in C1 complex? Calcium ion, C1S, C1R, C1Q. These are all are present. Remember this. Which type of protein or ingredient are present in C1 complex? C1Q, C1R, C1S and calcium ion. So the component required in classical pathway is C1 complex, C2, C3 and C4. Okay. Now, classical pathway generation of C3 convertase. Generation of C3 convertase. How the C3 convertase will be generated so that C3 protein will be activated, and after that C5 convertase will be generated so that C5 will be activated. So now we have first target is the generation of C3 convertase, right? So see, this is the C1 complex, right? This is a C1 complex. When this C1 complex will be activated, they activate the C2, right? C2, in C2, what happens? C2A will be removed and C2B will be present. Sorry. C2 is exception, right? Remember this C2 is exception. So this C2B is removed. C2B is removed and C2A will be remain for the binding. Clear? So first what happened? First C1 complex has C4, then C2, then C3B. Clear? So C4B, C2A and 3B is a C5 convertase. What? This is a C5 convertase and it leads into a membrane attack pathway. When this C5 convertase will be activated, they activate the C5. What? C5. Right? And after that, this C5 help in membrane attack pathway. Clear? Clear this one? See, this is the stimulation by antibody, especially IgM and IgG. Because in previous slide, already see that classical pathway is an antibody dependent pathway, right? So there are 5 subclasses of antibody. So this pathway is depend on which subclass, right? So this is a IgM. This depend on the IgM and IgG. Remember that. The classical pathway is stimulated by antibody, especially IgM and IgG. What? IgM and IgG. It starts with C1Q, a huge protein, 44 lakh 10,000 Dalton, composed of 18 peptide. 18 peptide. Right? Peptides can associate to form trimer. 6 set of trimer make C1Q. See here, this is a 6 set of 6 set of trimer that make a C1q, right? C1q has a helical stalks and globular. This is the globular head and this is a stalk, right? This is composed of 18 peptide, right? Right? So this is a C1q hues protein, right? That help in classical pathway. The head are the carboxy and the tail are the amino end. Means this is the amine group and this head is the carboxyl group. Okay, clear? Now see what happened here. See, this is the proper pathway of the classical pathway. Okay, how the C5 convertase will be formed. See, this is the C1. Where is the C1q? This is the C1q. In previous slide you see that this is the stalk, this is the globular head and this is amine group and this is the carboxyl group and so this is a C1 polypeptide. They activate the C4. First activate what? C4 will be activated. When C4 will be activated, they activate C2. When C2 is activated, they activate C3. Right? When C3 will be activated, they form a C5 convertase. When this C5 convertase will be formed, they attack as a membrane attack. What? Membrane attack. Clear? This is the when the C5 will be activated, they activate the 6, 7 like that, right? When 6, 7 will be activated, see here. When 6, 7 will be activated, this C2b is removed. C2b is removed and C2a will be remain for the binding. Clear? So first what happened? First C1 complex C4 then C2 then C3b. Clear? So C4b, C2a and 3b is a C5 convertase. What? This is a C5 convertase and it leads into a membrane attack pathway. When this C5 convertase will be activated, they activate the C5. What? C5. Right? And after that, this C5 help in membrane attack pathway. Clear? Clear this one? See, this is the stimulation by antibody, especially IgM and IgG. Because in previous slide, already see that classical pathway is an antibody dependent pathway, right? So there are 5 subclasses of antibody. So this pathway is depend on which subclass, right? So this is a IgM. This depend on the IgM and IgG. Remember that. The classical pathway is stimulated by antibody, especially IgM and IgG. What? IgM and IgG. It starts with C1q, a huge protein, 4,10,000 Dalton, composed of 18 peptide. Composed of 18 peptide. Right? Peptides can associate to form trimer. 6 set of trimer make C1Q. See here, this is a 6 set of 6 set of trimer that make a C1Q, right? C1Q has a helical stalks and globular. This is the globular head and this is a stalk, right? This is composed of 18 peptide, right? Right? So this is a C1Q hues protein, right? That help in classical pathway. The head are the carboxy and the tail are the amino end. Means this is the amine group and this head is the carboxyl group. Okay, clear? Now see what happened here. See, this is the proper pathway of the classical pathway. Okay, how the C5 convertase will be formed. See, this is the C1. Where is the C1Q? This is the C1Q. In previous slide you see that this is the stalk, this is the globular head and this is amine group and this is the carboxyl group and so this is a C1 polypeptide. They activate the C4. First activate what? C4 will be activated. When C4 will be activated, they activate C2. When C2 is activated, they activate C3. Right? When C3 will be activated, they form a C5 convertase. When this C5 convertase will be formed, they attack like as a membrane attack. What? Membrane attack. Clear? This is the when the C5 will be activated, they activate the 6, 7 like that, right? When 6, 7 will be activated, see here. When 6, 7 will be activated, they go like that and bind to C9. After that, they phagocytose. When poly C9 will be present, they act as a membrane attack complex. They act as membrane attack complex and they help in lytic cycle. And they destroy the pathogen. So, biological activities of classical pathway components. Components C2b, C3a, C3b, C4a and C4b. C2b, what is the activity? Prockinine, cleaved by plasmine to yield kinine which result in edema. C3a, anaphyloxin can activate basophils and mast cells to degranulate, resulting in increased vascular permeability and contraction of smooth muscle cell, which may leads to anaphylaxis. C3b help in opsonine, activation of phagocytic cell. C4a, anaphylotoxin, help in anaphylotoxin, and C4b help in opsonization. What? Help in opsonization process. Okay. Control of classical pathway components. C1 inhibitor. All components are regulated by C1 inhibitor, that is C1 C11H dissociates C1 and C1S from C1Q. C3A is regulated by C3A inactivator, that is C3A and carboxypeptidase. C3B is regulated by factor H and I. C4A is regulated by C3A INH, and C4B is regulated by C4 binding protein. So the A4B is regulated by C4 binding protein. Clear? Now components of mannose binding lectin pathway. What? Mannose binding lectin pathway. In what happened in this pathway? In this pathway, the component is mannose binding lectin is itself component, right? Mannose binding, and this is present on a bacteria. So in this mannose binding lectin, C2 as in classical, C4 as in classical, and MASP2 and MASP1. This is the component of the mannose binding lectin pathway. Now mannose binding lectin pathway. First what happened? Mannose binding lectin will be present, right? They activate the MSP1 and MSP2. After that, C4 protein will be activated. They convert it into C4A and C4B. C4B is the larger component, so it is the larger component, so it will remain, and C4A released in the environment. Now C2B is C2 will become, and they cleaved into a C2A and C2B, right? They converted into a C3 convertase. It will lead to generation of C5 convertase, right? When C5 convertase will be generated, they activate the B, P and D, right? So C3, this that is the mannose binding pathway. This is the mannose binding lectin is the mannose binding lectin C4, C2, C3 is the component of the mannose binding pathway. Now what is the component of the alternative pathway? The component of alternative pathway is the C3, B, P and D, right? In alternative system, C3 is the principal protein of concern, exists in high concentration, contain an unstable internal thioester with slow spontaneous or serum protease mediated cleavage. Clear? By convention, the smaller product is designated A and the larger is B. Already I told that a smaller one is A and larger one is B, except C2. Except C2. C3b can bind to bacteria and enhance phagocytosis. These are the alternative pathways. The production of C3b can be catalyzed by C3 convertase. C3b can be catalyzed by C3 convertase. This is the factor B, factor D. When they combine to form the factor, they bind to factor B. This factor bind with C3b, right? This binding of propidine stabilizes C3b and BB, C3b and BBB. This is the C3 convertase. What? C3 convertase. So what happened in alternative system? When C3 convertase will be activated, they activate the C5 convertase. When C5 convertase will be activated, the C5 convertase activate the C5, and already know that C5 will clip into C5A and C5B like that, and when C5B is unstable and is stabilized by C6. When C6 will be activated, allow C7 like that 8, and when 9 will be activated, they form a poly C9. When poly C9 will be formed, then they act like a membrane attack complex. In that, they phagocytose the foreign subparticles and they destroy from the body. Oh, clear? Now the complement pathway. See, this is the combination of all the complement pathway. This is the lactane pathway, this is classical pathway, and this is the alternative pathway. Lactane pathway, MBL is used. In classical pathway, antigen antibody is used, and in alternative pathway, microbial surface is used. So remember this all pathway. Most important in complement system is the pathway. So lactane pathway, mannose binding lactane is used. That is microbial cell wall. In alternative pathway, microbial surface is used and factor D is used. And in classical pathway, antigen antibody complex is used. Okay. So biological effect of C5A. C5A help in adhesion, chemotaxis, vascular wall transmission, mast cell degranulation, and cytokinin production. This is the function of the C5. Now, biological properties of C activation products. C2B prokinin, C3 anaphyloxy, and the biological effect of C2B is edema and regulated by C1NH. So this already I told you in previous slide. Complement deficiency and disease classical pathway. Pathway C1NH, C1, C2, C4. Disease is hereditary, angioedema, predisposition to SLE, mechanism overproduction of C2B, and opsonization of immune complexes help keep them soluble. Deficiency result in increased precipitation in tissue and inflammation. So this is all are the deficiency. What? Deficiency of the disease with of complement system in alternative pathway, classical pathway, and lactose binding pathway. Clear? So this all about the complement system. Today we stop here. Hope you understand all three pathways. So today we discuss about the humoral and cell mediated immunity that comes under serology and immunology unit. So first of all, what is humoral and what is cell mediated immunity? From name it's clear, right? From name it is clear that humoral immunity, the immunity that present from the birth or that are humoral immunity that our cell, our body will fight against the pathogen. Our immunity when fight like skin, like skin So here this all comes under humoral immunity. And cell mediated immunity means the immunity that mediated by cell like B cell, T cell. So in cell mediated, mainly two types of cell will become, that is B and T cell. Clear? What is the humoral and what is the cell mediated immunity? We discussed in this video about the humoral and cell mediated immunity in detail. So definition. So first of all, definition. Immune system. What are the immune system? The immune system, in our body, the system which fight against the infection, that known as the immune system, right? And what comes under this immune system? Which part of our body will fight against the infection? Like cells, tissue and molecules that mediate resistance to infection. So the cells and tissue like pear patches, tonsils, skin, hair, this all comes under our immune system. Clear? Next, immunology. What is immunology? Immunology is the study of the whole immunity. Study of a structure and function of the immune system. This is known as immunology. Next is immunity. Resistance of a host to pathogen and their toxic effect. Immune response, collective and coordinated response to the introduction of foreign substances in an individual mediated by the cells and molecules of the immune system. Collective and coordinated response to the introduction of foreign substances in an individual mediated by cells and molecules of the immune system. Clear? Role of the immune system. What is the role of immune system? Main role is the defense against microbes. Defense against the microbes. Defense against the growth of tumor cells. Kill growth of tumor cells. Homeostasis. Destruction of abnormal or dead cells. Homeostasis means destruction of all the abnormal or dead cells. This is known as the homeostasis. Immune systems. What comes under immune system? In immune system, our organ system comes organ, cells and molecules. In molecules like cytokinin, lymphokinin, this all are a molecule. Lymphokinin, right? Immune system. First organ that's come under immune system is tonsil, adenoids, thymus, lymph nodes, spleen, pear patches, appendix, lymphatic vessels, bone marrow. What? Tonsils, adenoids, thymus, lymph node, spleen, pear patches, appendix, lymphatic vessels and bone marrow. This all comes under immune system. Okay. Next is lymphocytes. What are the lymphocytes? Lymphocytes are T cell lymphocytes, B cell lymphocytes. When B cells will differentiated, they differentiate into a plasma cells and memory cells, right? When B cell will differentiated, they converted into plasma and memory cell, and plasma cell contain antibody, and memory cell will remember the pathogen for long time. And next lymphocytes is the natural killer lymphocytes. Next is monocytes, macrophage, granulocytes. In granulocytes is neutrophils, is eosinophils, basophils. Granulocytes contain neutrophils, eosinophils, basophils. Clear? Now third is antibody. Third is molecules. And in molecules what contain? Antibody, complement system, cytokinin, interleukin and interferon. interferon. Remember this. This all are a molecule. Antibody, complement system, cytokinin, interleukin and interferon. There are two types of immunity. One is innate immunity and another is acquired immunity. Innate immunity means the immunity that present from our birth, and this immunity is known as a first line of immune response. What? First line of immune response. And it relies on mechanisms that exist before infection. And next immunity is adaptive or acquired immunity. Acquired immunity means what happened? The immunity and the substance which acquired after birth from the environment, the immunity adapt by that process is known as a acquired immunity. And this adaptive immunity or acquired immunity is known as second line of response. When innate immunity that present from our birth is fail, after that acquired immunity will produce their effect, their response. So that this immunity is known as second line of response. Acquired immunity relies on mechanisms that adapt after infection. When infection takes place, first innate immunity will be activated. When innate immunity will be activated, and after that the infection will not cure, then artificially we take a drug, right? Or enhance our immune system, we eat something drug like that, so that our immunity will be increased. When antibody will be react as a second line of response, the B cell will be differentiated into two types, that is plasma cells and memory cells. So this memory cell will be always remember this type of immunity for a long time. When second time this same infection will be attack on our body, then immediately the second line of response, that is acquired immunity, will be activated and they kill that pathogen. So this is the mechanism. Acquired immunity handled by T and B cell. Which? T and B cell. One cell determine one antigenic determinant. Remember this. One cell determine one antigenic determinant. So first is innate immunity. Innate immunity based on genetic makeup. Already told you that innate immunity are present from birth. Innate immunity are present from the birth. Relies on already formed components, rapid response within minutes of infection. Innate immunity, what happened? Innate immunity having a rapid response. What? They having rapid response. When infection will attack on a body, the first line of defense is innate immunity. They activated in minutes. It is not a specific. Innate immunity is not a specific immunity. Same molecule say responds to a range of pathogen. Has no memory. Remember this. Has no memory like B cells or B cells. Does not leads to clonal expansion. Does not lead to clonal expansion. Innate immunity mechanism. Mechanical barrier. In mechanical barrier, what present? Skin acidic pH in stomach cilia. Humoral mechanism, lysozyme, basic proteins, complement interferons. And in cellular defense mechanism, natural killer cells, neutrophils, macrophages, Mars cells, basophil and eosinophils. These all are a innate immunity mechanism. What? Mechanical barrier, humoral mechanism, cellular defense mechanism. This is the innate immunity mechanism. Clear? Adaptive immunity, second line of response. Based upon resistance acquired during life. What? Based on the resistance that acquired during life, relies on genetic events and cellular growth. Responds more slowly over few days. What? They respond slowly. What? Innate immunity responds fastly within a minute, but this responds in a few days. Is specific. Each cell responds to a single epitope on an antigen. Response on a single epitope on an antigen. Has anamnestic memory. Repeated exposure leads to faster and stronger response. First, see, this is a graph. First response is a so late. But second response, see, second response is so fast. This is a latent period. In first response, this latent period is more. But in second response, this is a very less. So, second response is very fast. Leads to clonal expansion. They increase the number. In adaptive immunity, what happened? They increase the number. Adaptive immunity, active and passive. Adaptive immunity, active and passive. They are differentiated into active immunity and passive immunity. In active immunity, they are natural, clinical, subclinical infection, right? In passive, via breast milk and placenta. Passive immunity are provided by the breast milk and placenta. And artificially they are by vaccination, live, killed, purified antigen vaccine. So, these are the artificial type of active immunity. And passive immunity, an artificial type of passive immunity is immune serum, immune cells. What? Immune serum and immune cells. This is the type of passive immunity. Clear? Adaptive immunity mechanism. Cell-mediated immune response. First is cell-mediated immune response, that is T lymphocytes. Next is eliminate intracellular microbes that survive within phagocytes or other infected cells. And next one is humoral immune response. B lymphocytes mediated by antibody eliminate extracellular microbes and their toxin. Remember this, the mechanism of adaptive immunity is mediated by two types, that is cell mediated immune response and humoral immune response. So, cell mediated immune response, that is by your T lymphocytes, and humoral immune response, that is of B lymphocytes, right? So, B lymphocyte mediated by antibodies eliminate extracellular microbes and their toxin. Eliminate extracellular microbes and their toxin. Adaptive immunity, that is active and passive. Already I told you active and passive immunity. So, first is adaptive immunity. Adaptive immune system has two arms, that is humoral immunity and cell mediated immunity. So, in starting I told you that humoral cell mediated immunity is controlled by B and T cells. So that's not, in starting I tell you wrong, that humoral immunity is controlled by B cell and cell mediated immunity is controlled by T cells. Clear? So remember this, humoral immunity controlled by B cells and cell mediated immunity controlled by T cells. Humoral immunity can recognize protein, polysaccharide, phospholipids and nucleic acid antigen. And cell mediated immunity can recognize only protein antigen. Clear? Cell mediated immunity recognize only protein antigen, whereas humoral immunity can recognize protein, polysaccharide, phospholipid and nucleic acid antigen. Humoral immunity can act against soluble or free antigen. Cell mediated immunity recognize antigen presented by APC with class 1 or class 2 MHC molecule. Humoral immunity provides immunity to extracellular bacteria, virus and toxin. Extracellular bacteria, virus and toxin, and causes type 1st, 2nd and 3rd hypersensitivity. Which type of sensitivity they humoral immunity? 1, 2 and 3. Right? And cell mediated immunity provide type 4 hypersensitivity. Okay? And cell mediated immunity cause acute graft rejection. Clear? Now cell mediated immune response. Cell mediated immune response is mediated by T cells, right? So what happened? T cell recognize peptide antigen. What? They recognize the peptide antigen on macrophages in associated with major histocompatibility complex, that is MHC. MHC is of two types, MHC 1 and MHC 2. Actually T cells is of two types, TC cell and TH cell, right? TH is T helper cells and TC cells, right? For these two type of MHC are present, MHC 1 and MHC 2. Identify molecules on cell surface, help body distinguish cell from non-cell. This MHC distinguish between self and non-self. So T cell goes into effector cell stage, that is able to kill infected cells. T cell goes into effector cells stage, that is able to kill infected cell. This is the T cell activity. See, this is cytotoxic T cell, TC cells. This is the TC cell, right? This is antigen presenting cell, APC, that is called antigen presenting cell. When antigen recognized by antigen presenting cell, this T cell, cytotoxic cell, will came and bind with this, right? In third step, what happened? This helper cell came and bind with this, they activate the T cell. They activate the lots of T cells, TC cells, so that they produce lots of molecules. In starting already I told you molecules, cytokinin, antibody, interleukin, this all are comes under molecules, right? So that this when TC cell will be activated, they release release lots of molecules, that is cytokinin. And this cytokinin degrades this pathogen. What? They degrades the pathogen. Clear? Clear? And this one step is also that they activate the B cell. This helper cell activate the B cell, and this B cell produce antibody, and this antibody degrade the APC molecule, antigen presenting cells. Right? So these are the mechanism of T cells. What? These are the mechanism of T cells. T lymphocytes. T lymphocytes are of two types. Already I told you. T lymphocytes are of two types. That is T helper cell, TH, and T cytotoxic cell. That is TC. This TH cell having CD4 T cells activate phagocytes to kill microbes, and CD8 plus present on TC cells that destroy infected cells containing microbes or microbial protein. They destroy infected cells containing microbes or microbial protein. Now, this is the antigen, right? In antigen, what are present? Foreign protein, virus, bacteria. What? Foreign protein, virus, and bacteria. This antigen having two types of response. Having two, that is humoral response. They create humoral response or cellular response. If humoral response are present, the B cells will be activated. If cellular response will be present, T cell will be activated, right? When B cell activated, they differentiated into two types. That is plasma cells and memory cells. Another is BM, memory cells, right? Plasma cells produce antibody. They secrete antibody, and this antibody attack on antigen so that they kill that antigen. And next one is cellular response. That is T cells. When T cell recognize, they differentiate into TC and TH cells. TH cells produce cytokinin, or they help to activate B cell, right? And TC cell kill the infected cells. They kill the infected cells by producing the molecule like cytokinin or interleukin like molecule. By that the infected cells will be killed. These are the mechanism of cell mediated immunity. Now, cell mediated immune response. Primary response, production of a specific clones of effector T cells and memory clones. In primary response, what happened? Production of specific clone. The production of a specific clones increases in primary response. Developed in several days, does not limit the infection. Secondary response, more pronounced, faster. Already I told that in primary response, what happened? They take several days for recognizing or fight against that infection. They take several days in primary response or first response. But when memory cells will developed in the body, in second response, they more effective, and they recognize that same infection very quickly. So secondary response is more pronounced, faster, more effective at limiting the infection. An example is cytotoxic reaction against intracellular parasites delayed hypersensitivity, hypersensitivity, tuberculin test, and allograft rejection. Now, next is humoral immune response. Humoral immune response is a B lymphocyte recognize a specific antigen. B lymphocytes recognize a specific antigen that proliferate and differentiate into antibody secreting plasma cells. What? They proliferate and differentiate into antibody secreting plasma cells. Antibody binds to a specific antigens on microbes, destroy microbes via a specific mechanism. They destroy microbes by a specific mechanism, and some B lymphocytes evolve into the resting state memory cells. What? In resting state memory cell, because B cell will differentiate it into two types, right? In plasma cells and in memory cells. And plasma cells will produce antibody, whereas memory cells recognize the antigen when second time they infect our body system, right? Now, these are the mechanism of the humoral immune system. This is the antigen. These are the antigen, right? This is the antigen specific B cell receptor. Antigen specific B cell receptor class 2 MHC will be activated, right? Class 2 MHC will be activated. They recognize by the class 2 MHC. This antigen is processed via MHC, right? After processing, class 2 MHC and processed antigen are displayed. They activate helper T helper cells. After activating helper T cell, they activate the B cell. When B cell be activated, they differentiated into two types, plasma and memory cells. This plasma cell produce antibody. This antibody kill this antigen. So these are the process or mechanism by which humoral immunity will be takes place. Now, antibody. What are the antibody that are produced by the humoral immunity? So, antibody, also known as immunoglobulin, belongs to the gamma globulin fraction of serum protein. It is a Y-shaped structure or T-shaped polypeptide. They having two identical heavy chain and two identical light chain, and these two heavy chain and light chain are attached by a disulfide bond. All immunoglobulin are not antibody. Remember this. 5 kinds of antibody are present, that is IgA, IgM, IgE, IgD and IgG. IgG having a 70 to 75% of total immunoglobulin and secreted in high quantities in secondary exposure, and this is the only type of immunoglobulin that that cross the placenta, and these immunoglobulin are present before the birth in a child. Major function of this is to neutralize microbes and toxin, opsonize antigen for phagocytosis, activate complement system and protect the newborn. IgM secreted initially during primary infection. When first infection attack on a body, then the level of IgM will be increased. But in second time, level of IgG will be increased. Remember this. IgM cannot cross the placenta. Major function: secrete first during primary exposure, activate the complement, used as a marker of recent, and IgA is monomeric in serum, dimeric with secretory component in the lumen of the gastrointestinal tract in the respiratory tract. Major function, neutralize microbes and toxin. IgD is the monomeric. Major function, present on surface of B lymphocyte, function as membrane receptor. Main role of IgD is unclear. Ig is a hypersensitivity, monomeric, major function associated with anaphylaxis and play in immunity helminth parasites. Right? Sequential IgM IgG humoral response. IgM is the primary response. IgG is the level of IgG will be increased when in secondary response or produced after the IgM. Higher level processes smaller amount throughout life, produce a larger amount during secondary response. Already I told this. See, this is the graph of IgM and IgG. See, this is the level. In first response, the level of IgM will be increased in first response, and in second response the level of IgG will be increased. Right? Failure of immune response. Immune response help individual defend against microbes. Some cancer immune response can fail. Hypersensitivity reactions, immunodeficiency. Hypersensitivity reaction cause cells damage through excessive immune response to antigen. Hypersensitivity overreaction to infectious agent, allergy, autoimmunity. Clear? Immunodeficiency, loss or inadequate function of various components of the immune system can occur in any parts or state of the immune system. The immunocompromised host has an impaired function of immune system. Immunodeficiency means impaired function of immune system. It is of two types, congenital and acquired. Congenital means primary immunodeficiency and acquired by secondary immunodeficiency. Congenital is genetic abnormality, where acquired is the result from infection, nutritional deficiency or treatment. Cytokinin is a molecule that's secreted by T cells or B cells, right? And they help in destroying the pathogen. Types of cytokines: monokines, lymphokines, interleukin, chemokines, right? So this all about the humoral and cell mediated immunity. So hope you understand this. So I stopped here. Thank you.